Ever wondered about the importance of monoclonal antibodies in medical research? Picture this. They're like specialized agents, each designed with a unique mission to identify and bind to specific proteins or antigens. This ability to target specific proteins is what makes them so valuable. Monoclonal antibodies are used in a wide array of medical applications. They're key players in diagnostics, helping us detect the presence of disease markers. In therapeutics, they're used to block harmful signals from disease-causing antigens or to deliver drugs directly to disease cells. They're also crucial in the study of molecular and cellular biology, allowing researchers to probe the inner workings of cells and unravel the mysteries of life itself. So it's clear that these microscopic marvels play a vital role in advancing our understanding and treatment of diseases. But you may be wondering, these agents don't just appear out of thin air. Now that we understand their importance, how are these monoclonal antibodies produced? The process of monoclonal antibody production starts with a specific antigen. This antigen is like a puzzle piece, unique in shape and size, and it's our job to find its perfect match. Once we have our antigen, we need an animal model, most commonly a laboratory mouse, which becomes our key partner in this scientific endeavor. We introduce this antigen to our little helper, essentially immunizing the mouse. This is not a one-time event. Oh no, this antigenic initiation is repeated multiple times. Why, you ask? Well, we want to make sure the mouse's immune system really gets to know this antigen, really understands it. The B cells of our immunized mouse are like diligent students studying the antigen, learning its every curve and corner. These cells are then removed from the spleen, ready to embark on the next phase of our journey. Once the animal is successfully immunized, it's time to move to the next step. The next crucial step is the fusion of B cells with myeloma cells. We're diving into the heart of the process. The B cells of the immunized animal are carefully extracted from the spleen and then fused with a special type of cancer cell, the myeloma cells. Imagine this fusion as a merging of the best characteristics from both cell types. The B cells carry the specific antigen response, while the myeloma cells hold the key to immortality, continuously proliferating. But there's a catch. The normal B cells only proliferate for a period of 6 to 8 hours. That's where the myeloma cells step in. These cancer cells have lost the ability to synthesize an enzyme necessary for the degradative synthesis of nucleic acids, known as hypoxanthine-guanine-phosphoribosyl transferase, or HGPRT for short. This loss is actually a gain for us. The fusion of these B and myeloma cells results in hybrid cells that not only carry the desired antigen response, but also have the ability to grow indefinitely. The fusion process is facilitated by a compound called polyethylene glycol. However, it's not always a guaranteed success. That's why these fused cells are then placed in a selective growth medium, the hypoxanthine-aminopyrimidine, or HAT medium. This medium is a cocktail of hypoxanthine, aminopterin, and thymidine. It's designed to promote the growth of only the fused cells, the hybridomas. The non-fused myeloma cells lacking the essential enzyme HGPRT can't grow in this medium, and the non-fused B cells, they simply die off. In this selective medium, the hybridoma cells thrive. They grow continuously, churning out the specific antibodies we are after. These cells are essentially tiny factories, working tirelessly to deliver the valuable product, the monoclonal antibodies. The selective medium not only promotes the growth of fused cells, but also ensures the growth of only desirable cells. That's the beauty of this fusion and selection process. It's a meticulous dance of science that leads us to our goal, producing specific, plentiful monoclonal antibodies. With our hybridoma cells growing, it's time for screening and harvesting. As our hybridoma cells continue to multiply and flourish in their culture, we're ready to move on to the next stage, the screening process. This is where we look for those unique cells producing the monoclonal antibodies we want. Remember, these antibodies are specific to the antigen that was initially injected into our animal model. Screening is a meticulous process that involves testing each hybridoma clone to identify the ones producing the desired monoclonal antibodies. This is typically done using an ELISA, or enzyme-linked immunosorbent assay. The ELISA will detect the presence of the antigen-specific antibodies produced by the hybridomas. Once we've identified the cells producing the monoclonal antibodies we're after, these specific hybridoma cells are transferred and allowed to grow in tissue culture. This selective process ensures that we are cultivating only the cells that will yield our desired antibodies. So, what happens next? The process of harvesting begins. This is where the magic truly happens. The monoclonal antibodies produced by the hybridoma cells are secreted into the culture medium. From there, they're periodically collected over several weeks. But our job doesn't end there. These harvested antibodies are not yet ready for use. They must first undergo purification. This process involves removing any unwanted components from the medium to ensure we're left with only the pure, specific monoclonal antibodies. This entire process, from screening to harvesting, is repeated several times over a span of weeks. This is done to produce enough monoclonal antibodies that can be used for experimentation or to treat at least a single patient. The end result? Millions of monoclonal antibodies, all specific to the antigen that was initially injected into the animal model. These antibodies are now ready to play their part in a variety of medical applications, from diagnostic tests to targeted therapies. And there you have it, monoclonal antibodies, ready for use in a variety of medical applications. Let's take a moment to recap our journey in the production of monoclonal antibodies. We began with the identification of a specific antigen and the immunization of an animal model, usually a laboratory mouse. The B cells of the immunized animals were then removed from the spleen and fused with myeloma cancer cells. Despite the low success rate of this fusion process, the use of a selective medium, hypoxanthine-guanine-phosphoribosyl, or HGPRT, ensures the growth and yield of the desired hybridoma cells. These hybridoma cells, given their capacity to proliferate indefinitely, are then screened for the production of the desired monoclonal antibodies. Once identified, these cells are grown in tissue culture, and the monoclonal antibodies are harvested periodically from the medium. This process is repeated for several weeks to produce enough monoclonal antibodies for experimentation or treatment. So the next time you hear about monoclonal antibodies, you'll know exactly how they're made and why they're so important in medical research. Until next time, keep exploring.